Hello. Next chapter, we'll discuss about what are different gadgets used in total quality management in IVF. Okay. So for IVF, we need good amount of temperature, good amount of pH, osmolality, how should be the environment, air quality. Okay. Cleaning procedure. Those things we'll learn. Okay. So first, temperature. We all know temperature fluctuations are there, and the meiotic spindles, whatever meiotic spindles are here, they are very sensitive to temperature fluctuations. If there is any temperature fluctuation, then it results in aneuploidy, disruption of embryo, implantation difficulty, and decreased pregnancy rate. Okay. So one of the most crucial areas for temperature loss is incubator, where we'll keep embryos. So embryos are kept in one incubator. No? That is the area where there is temperature loss. Okay, so when the lid is open and again the temperature to come to the normal standard temperature recovery time, it takes time. For example, if there is a large incubator with multiple doors, then the recovery time takes about 30 minutes. Okay, so if there is increased door openings, there is temperature loss and increased miscarriage rate. That is one point. Second place where temperature fluctuations are quite common is micro manipulator. Okay, so, one study in Cambridge, it shows us says that there is a quite significant temperature loss which occurs during ICSI if caution is not observed. Okay, while doing ICSI only, there will be more temperature loss. Okay, so if you see spindle imaging with objective heater, without objective heater, if there is without objective heater, then the spindle imaging, if you see ICSI, there is only 61% of pregnancy rate. Whereas compared to objective heater, it will be good. Okay. If there is objective heater, then the spindle imaging, spindle formation will be very good. Okay. They also compared outcomes from various heating staging, like heated stage with aperture, heated glass stage, heated stage with aperture, and air warming system. These are three heating stages. Okay. Heated stage with aperture, heated stage with the glass stage, heated stage with aperture, and air warming system. So they compared these three and they found that this air warming system has most stable temperature. Okay, if you see this image, there is spindle imaging. You can see spindle formation here. Okay, there is a spindle formation here, whereas there is no spindle formation in the down image. So when there is spindle imaging, it is a heated stage. Okay, without heated stage, there is no spindle imaging. Okay, that's about spindle. Coming to pH. pH is vital for embryo development. Okay, more often it is very vital for oocyte viability. Okay. Um, So oocytes are they can't tolerate the pH fluctuations. Okay, so very important is we need to monitor the pH. So sometimes they'll give this image with a box and one more box, okay, which indicates the pH. So pH should be maintained at least in a range of 7.3. Okay, like it nearly 7.3. Like for fertilization, it is 7.3. For blastocyst, it is 7.3. Only for cleavage, it is 7.2 in that way. So that range should be maintained. Okay. Hmm. So nowadays there are good incubators, new incubators with the option of online pH monitoring, which give a accurate, reliable time, efficient way for monitoring the pH. That's about pH. So temperature we learned, pH we learned. Now we learn about osmolality. So obviously, osmolality if it is more than 310, how much more than 310, then there is effect on embryo development. Okay, so osmotic changes. We should take precautions so that there will be no osmotic differences. Okay, like properly we should do dish preparation techniques, humidified incubators, oil overlay, etc. Okay, so osmometers are available to monitor the osmolality, but it is a very tedious process. So it's a great tool to monitor osmolality when preparing media in-house. But when buying standardized media, proper precautions eliminate the need for regular osmolality. So if we build that media in our own laboratory, then we can maintain that osmolality. Instead of buying the media from outside, then we should compulsory check with the osmometer before transferring the embryo. That's about osmolality. So more osmolality, then there is decreased implant. There is decreased pregnancy rates. That's about osmolality. Now coming to fourth important point in IVF lab is environment. Cultural environment and lab environment are next big things to check. Okay. Hislop conducted a study. He says that between a unique chain of enclosed workstations with incubator set at air temperature of 35 degrees and hot plate of 37.2 degrees, That is one given, and an open stereo microscope in conventional glass to cabinet with heated surface of 37.4 degree. Okay, one one is incubator with temperature and CO2 control with temperature of 35 and hot plate of 37.2. Other is open stereo microscope where the temperature is maintained as 37.4. These two he compared. Okay, so comparison indicates that there is a fluctuation in environment and temperature. Then there is uh, he concluded that if you keep an incubator like condition, then the fluctuations will be less. Okay, I don't know, in incubator like condition, if you keep the air temperature of 35 and you keep different hot plate of 37.2, then that will be helpful to maintain the embryo formation good. Okay, that's about temperature. Coming to air quality. Air quality means one study like GROW have noted that effect of declining the air quality on reproductive outcomes after IVF are variable and increased see nitric oxide if it is increased and it is consistently resulting in lower birth rate. Okay, air quality depends. Fine particulate matter. This is see what is PM particulate matter. Fine particulate matter of 2.5 in the IFF laboratory during embryo culture was associated with decreased consumption rate. Okay, so if fine particulate matter are increasing, then there is consumption rate is decreased. And United States Environmental Protection Agency (US EPA) confirmed that there is significant risk to the humans and fertility following exposure to some chemicals like wax and particulate matter. If the embryo is exposed, if the embryo is exposed to these chemicals, then there is significant risk for the humans and fertility. Okay. So that's why they keep some carbon activated carbon filters and potassium permanganate filters to clear these walks and PMs. Okay. Mm, and that's it. Okay. So mainly an embryology lab should be very clean. There should not be any dirt, dust, smoke, etc. Okay. Mm, that's why so many IVF laboratories have this HEPA filtration. Um, but HEPA can only remove the infectious agents and particulate about 0.3 micron. Okay. But these WOX and PMs are they are very small particles. To remove that, we should keep activated carbon filter and potassium permanganate filter. Okay. Media and consumables. Quality gadgets doesn't just cover the ART laboratory, but also extend the supply department. So there will be high quality gadgets should be available. So all the things I told about temperature, pH, environment, osmolality, okay, these things. How to clean these things, how to clean the gadgets, okay. We are pretty much moved away from alcohol being used in the laboratory, and now we are using the quaternary ammonium compounds, right, which are walk-free and quite reliable. But some of us are going back to use steam cleaners, which are quite effective, walk-free, safe, and easily available technique in the lab. So nowadays they're using steam cleaners. So if the options it is make potassium, sorry, ammonium compounds, you can keep that also, or the steam cleaner is also better. Okay. If you see this FertiSafe Plus spray, which is a walk-free compound. Okay, what is walk? Once again, a little bit. What is walk? Walk is volatile organic compounds. Okay, good. Safety and security is very important. Embryology labs have different risks because of storage risks, mix-ups, and staff, patient safety, all those things. So we should keep alarm systems in case of incubators and liquid nitrogen that it is gone or it is done or it is already been infected. That way, if you keep alarm system, then you can easily document the incidents and report the concerned staff immediately. Okay. Conclusion of this chapter is we have definitely entered the age of automation, and there is a variety of gadgets available to us which can ensure better quality of care. The only problem that arises is cost effectiveness of such solutions. In government funded setups, buying such equipment is easy, but in private clinics, such equipments can be huge financial investment burden. Also, utilizing these will directly as well as indirectly increase the cost for the patient, which in country like ours is not feasible. So, in this age of optimization, we need to find a cost-effective way if offering the best quality without compromising on the standard of patient care and still running a viable clinic. It's about fancy gadgets. Okay. Expected question in this chapter is, I feel this one. If there is heated state, with heated state there is spindle formation. Without heated state there is no much spindle formation. That I'm expecting this question. Okay. pH also you should want. Next chapter is absolute stenozoospermia. Absolute stenozoospermia. So it is a common cause of male infertility. See, steno means there is no motility. Okay, there is reduced motility or absent motility in fresh ejaculate. That is very important. Okay, not the old sample. It is fresh ejaculate. There is no motility or decreased motility. Okay, absolute means absolute stenozoospermia means there is 100% there is immortal spermatozoa. Frequency is in 5,000 men, one man will have this kind of condition. Low fertilization rate after ICSI with randomly selected immortal spermatozoa, especially when using ejaculate spermatozoa. Okay, you're taking the ejaculate spermatozoa and you tried the ICSI. Even then, there is no fertilization. That means even then the even though we kept in cytoplasm, it is could not able to travel till nucleus. Okay, that is absolute stenozoospermia. Why there is absolute estenozoospermia? What is the cause? It is because of high FSH. Okay, high FSH. Why there is high FSH? Why brain is producing high FSH means down, down below the gonads are not producing proper testosterone. If there is testicular failure, then the brain produces high FSH. Okay, and if you see flagellum of the sperm, you can see ultrastructural defects in the flagellum sperm, which indicates immortal but variable sperms. They are good live sperms, but they're immortal sperms. Immortal cilia syndrome is also there. It's an autosomal recessive condition. It is an autosomal recessive condition where there is immortal microtubules of ciliated cells and spermatozoa. Absent in arms or abdominal microtubules are seen in case of immortal cilia syndrome. Immortal sperms with situs inversus. If there is situs inversus, then we should think about Carter Jenner syndrome. What is necrozoospermia? It is 100% dead sperms. Okay, because of ROS, ROS means reactive oxygen species, they penetrate the cell and induce degenerative processes in the nucleus, resulting in DNA damage. Genital infection, oxidative stress, cryopreservation, metabolic disorders, environmental pollutants exposure, and delayed epidermal transport also one of the reasons for asthenospermia. Hypogonadotrophic hypogonadism. This is also another. See, one is high FSH. Sometimes there will be low FSH. That's why there is low testosterone. So hypogonadotrophic hypogonadism also results in asthenospermia. Prolonged period of ejaculation. Okay, prolonged you're doing ejaculation, then also there is asthenospermia. And last final cause is anti-sperm antibodies. So these are different reasons for asthenospermia. One is high FSH, low is low FSH, then is immortal cilia syndrome, carcinogenesis syndrome, necrozoospermia, other genetic environmental pollutants and cryopreservations, and there is prolonged period of ejaculation and anti-sperm antibodies. How to diagnose asthenospermia? Okay, first thing is we should examine the semen sample at 37 degrees. What is 37 degrees? It's a room temperature. So we should examine at that temperature, not high, not low. So proper abstinence before because lower temperature may affect the motility and rarely may cause immobilization. So if you keep at 25 degrees or 24 degrees, it can cause immobilization, and if false negatively, we diagnose it as asthenospermia. Important to assess the second semen sample also is very important, meaning this is immortal cilia syndrome, where there's family history and transmission of electron microscope shows absent dynam. Okay, if there is necrozoospermia, vitality testing. If there is dead, they're immortal and also dead. Then also we should go for second sample. Anti-sperm antibodies, we should mix the antiglobulin reaction test and immunobead test. Infections like chronic prostatitis, urogenital examination is very important. Culture and transrectal ultrasound is very important in case of chronic prostatitis. Exposure of ejaculate to cold, spermicides, medical history. Okay, medical history, if there is any spermicide intake, spermicide application, then also it results in asthenospermia. Hypogonadotrophic hypogonadism, how you'll find out? By means of hormonal analysis. Okay, this is the investigation. Now, how you'll treat? How you'll treat a patient with asthenospermia? Treat the cause. Okay, so basically the sperm is unable to move. So for that we'll go with ICSI. We will go for ICSI after sperm selection. Testicular sperm extraction may also be considered in the men with normal testicular function whenever only limited proportion of ejaculated sperm are left because TESI will yield more viable sperm. Okay, TESI is very important. Testicular sperm extraction may be considered in the woman, sorry, in the men with normal testicular function. Okay, so if you get a condition of asthenospermia, It's a normal testicular function. What will be your treatment? It is not ICSI. It is testicular sperm extraction, followed by ICSI. Okay. Coming to host test. What is host test? You'll take the live spermatozoa. You'll take live spermatozoa, and you'll check the function of the membrane. How? You'll expose the spermatozoa to hypoosmotic conditions. Once there is a hypoosmotic condition, then there is swelling and curling of the tail due to water influx. Okay, swelling and curling of the tail due to water influx. It results in that means that is good live spermatozoa. So what is that hypoosmotic? It is 50% isotonic and 50% distilled water. This was used for selecting the sperms. That is host test. Next is mechanical touch technique. This means there is ICSI pipette, okay? There is ICSI pipette, and we'll touch the sperm with that pipette, okay? We'll touch the sperm with that pipette. Once you touch the sperm, the sperm tail will coil, okay? That indicates it's a mechanical touch technique and it is a live spermatozoa. But this reliability all depends upon the embryologist. If embryologist feel that the tail is being curled, then it is live. Some people will say no. That way, it depends. It depends on what different, okay? Um, next is if you see this electron microscope sperm axonyms, it is a normal axonym. Hmm. So if you see the figure A, it is normal axonym. You can see the dynams and these things. If you see second picture, it's an electron microscope, which shows absence of outer and the inner dynams. Okay, there outer and inner dynams are absent, which are 100% immotile spermatozoa. Last picture is it is showing 9 plus 0 syndrome. Okay, there is absence of um, uh, there is absence of axonyms itself. Okay, so sometimes we'll give a picture of this. If you see properly, you can see the it's a normal axonym. Okay, you can see dynams. This is outer dynam and this is inner dynam. These are outer dynams, these are inner dynams. Okay, these kind of dynams are absent in case of this. Okay, and if you see 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 dynams are there. Okay, if you see, and in between there is also center axonym, right? But here there are these things and there is absence of that thing. That is known as 9 plus 0. So they'll give an image of like this and they'll ask what is this? It is 9 plus 0 syndrome. Okay. So now we'll see the flow chart how to work up a patient with asthenospermia. If a male patient came with like couple came and with male patient have a report of asthenospermia and it's absolute asthenospermia. What is absolute? 100% there is immortality. Okay, 100% there is immortal sperm. Okay, and there is low fertilization rate. They already tried ICSI, but there is no fertilization rate. So how to work up that kind of patient? First thing is with one semen sample, if there is asthenospermia, you will go for second semen sample. Okay. In second semen sample also there is 100% immortal sperm. Okay. First we'll learn about all positive findings. Okay. If there is really there is 100% immortal sperm, then you will assess the viability. I told na you will see sperms are not motile. You will see whether they are live or not. You will assess the motility viability. If they are all dead, 100% which are immotile, they're also dead. 100% dead. Yes, that is known as necrozoospermia. Then you will perform TESI. What is TESI? You will test the hormones and you will directly extract from the testis. You directly extract. Okay, testicular extraction of sperm extraction. Okay, that is TESI, testicular sperm extraction. You will remove that sperm from the directly from the testis and you'll go for ICSI. Okay. In ICSI also, if there is motile sperm or not, you'll see. If it is motile sperm, then you can go for ICSI. Even after TESI also it is not motile, then you select the viable spermatozoa for ICSI. Okay, like host test, mechanical test, pentoxyphilin test, laser assisted sperm selection. Pentoxyphilin, laser assisted sperm selection. Okay, by the means of different tests, you'll find out, yeah, this is good sperm. You select that and you'll go for ICSI. If that in TESI only, if after TESI, if every sperm is motile, then you directly go for ICSI. No need of testing the viable sperm. That's about positive findings. Okay, if a male person with estenozoospermia, you repeated the semen sample and repeated sample have good sperms. There is no 100% immortal sperms, are good sperms, then nothing to do. Okay, if in 100% immortal sperms, if you check the viability, if there is no 100% dead sperms, only immortal, they are not mortal, but they're alive. If like that, then you should see they're immortal, they cannot move, but they're live. Okay, but they're live. They cannot move. So you should rule out infections. You should rule out any prolonged abstinence, any latex condom exposure. These causes that immortality. Okay, prolonged abstinence also it causes immortality. Then there are no such history or there is no history of infection. Then you can go for ultra structural defects. Okay, nowadays they are doing this ultrastructural defect, that is DNA fragmentation test, right? So you not DNA fragmentation, you will see the structure of the sperm, in fact. Okay, if there is structural abnormalities, if yes, there is structural abnormalities, again you will go for selection of viable sperm followed by ICSI. If there is no ultrastructural abnormalities, no, then it is unexplained absolute estenozoospermia. That means there is no electrostructural defect, there is no infection, there is no exposure of any latex or prolonged abstinence. Okay, in that way. So that is unexplained estenozoospermia. Uh, I told another two tests for selecting the viable sperms, right? One is pentoxifilin, other is laser-assisted sperm selection. So what is pentoxifilin? It improves the it improves the sperm motility by increasing the cyclic AMP. So sperm you add this pentoxifilin one and it improves the sperm motility because of cyclic AMP. Short exposure of the sperm to pentoxifilin followed by sperm washing prevents embryo toxicity. Next is laser-resisted sperm selection. Variable spermatozoa shows curling reaction of the tail to application of laser shot close to the tip of the sperm. So you will apply a laser to the tip of the sperm and there is curling. Okay. And what type of laser system you'll use? It is a non-contact 1.48 mm diode laser. Okay. Next is birefringence polarization microscopy. Polarization microscopy shows viable sperms as having birefringent head and midpiece, and using these improves the. If you see that birefringent thing, that means good sperms. If you select that, then there is a good pregnancy rate. So in this chapter, I feel that this image is very important. One thing they can give this image and ask you what is the syndrome, that is 9+0 syndrome. Another is this flowchart. If there is absolute testosterone and absolute necrozoospermia, and if you perform TESI and their motile sperm, then directly you can go for ICSI. And even not TESI, there is no motile sperm, then you'll go for sperm selection. Okay, they can ask this question. Next chapter. Now I'm saying we'll select sperms. Select sperms. So how we will select sperm? What are the different advances in sperm selection? Okay. Mm, so, basically, a natural process of sperm going from the vagina into cervix, into uterine cavity, into the fallopian tube, and reaching till ovary. In this travel pathway, there are so many physiological barriers, like acidic pH of vagina kills sperm. Cervical mucus doesn't allow sperm to go inside. Okay. Other coming to uterus, there is uterine environmental changes. Okay, coming from uterus to tube, uterus have different kind of environment. From uterus to tube, again, environmental changes occurs. So there also so many sperms die. Okay. Finally, sperm bind to zona pellucida. So there are so many barriers to the sperms. Okay. So to cross all these barriers, we should have a perfect and capable sperm, right? So all motile sperms do not have a potential. So if you think, if you see the sperm examination, semen examination, they say motility is this much, 90% motility, 80% motility. If they say that means that are not all good sperms. All motile sperms do not have a potential to fertilize the oocyte. So judging the sperms based on motility alone is not a best way to select. Okay. So now logically we try to mimic the natural selection IVF laboratory. So how bodies naturally selects now in that way in IVF laboratory we select the sperm. Good sperms we select. Okay. So head carries the DNA content. We all know head, midpiece, and tail. Okay, so head contains the DNA content. This DNA content should be delivered to the egg. Okay, and midpiece stores the energy. Midpiece stores the energy, creating a mitochondria. And tail is for movement, flagellar propulsion, and that way it go. So what are different techniques? The current techniques based on selecting the sperm is based on motility. In motility also there is swim up technique, and based on morphology, that is density gradient technique. But these do not really mimic any natural process. Okay, in natural process, in natural way of sperm going up, there is no swim up or density gradient things. It will go naturally. Okay, but the sperms which are selected by the embryologist for ICSI. ICSI manipulated only on that only they will select, and they do the selection process. But again, these modalities are only based on judgment of embryologists. So these are good sperms, these are good motility. All depends upon individual. It is a subjective decision. Okay. So the newer techniques which have come up to show the positive impact as far as results are concerned. PIXI or hyaluron binding capacity, intracytoplasmic morphology selected sperm injection. Till now we know ICSI. Now there is a another word known as IMSI. Okay, what is IMSI? Intracytoplasmic morphologically selected sperm injection. That is been evolving nowadays. Okay. So, intercytoplasmic morphologically selected sperm injection, microfluidic, magnetic activated sperm sorting, and omics. In this way, we can segregate good sperms. Okay. Uh, okay. Coming to hmm, Okay, coming to physiological ICSI. Physiological ICSI, like the sperm has a plasma membrane. Sperm has a plasma membrane. So plasma membrane remodeling is very important in preparing the sperm to fertilize the egg. During this process, specific receptors form like zona pellucida receptors and hyaluronic acid receptors, which aid in cumulus binding. The principle behind the use of PIXI is that when mature sperm reaches the hyaluronic acid rich cumulus complex surrounding the human egg, it binds. So, you understood now? So, egg, if it has a very good amount of hyaluronic acid, then this mature sperm go and bind to that egg. Okay, bind to the egg. It all depends upon hyaluronic acid. So, only few mature sperms have developed hyaluronic acid receptors. Okay, it should have some receptors. It go and bind to that hyaluronic acid. So, it can bind to the zona pellucida. So immature sperms cannot bind to hyaluronic acid and found to have higher rates of DNA damage and higher occurrence of abnormal number of chromosomes. Okay, so if there are immature sperms, but then they go and bind to the ovum and they form embryo which has abnormal number of chromosomes. Thus, the patients have high DNA fragmentation index or have suffered a recurrent miscarriage or have poor quality of embryos. For these people we can select good sperms because immature sperm we should not allow immature sperms to bind, right? So for them selection and then going for fertilization is very good option. Hmm. Coming to IMSI. What is IMSI? Intracytoplasmic morphologically selected sperm selection. So they have found that in case of IMSI, gross evaluation of sperm on ICSI magnification 400x times is not sufficient because see we need to see the sperm very properly. Totally, it is very okay. Then you can select it for IMSI. But even on ICSI magnification, that is 400 times magnification also, there are subtle aberrations. Even though if you see on that, sperm will be a very perfect sperm. Even then there are subtle aberrations and there is higher chances of transmitting the genetic as well as chromosomal diseases. So sperm vacuoles have been researched. Mm, so if sperm have vacuoles inside that head, it has high DNA fragmentation index, which this association, they're saying that if sperm have vacuoles in the head, there is more DNA fragmentation index, more aberrations, more chromosomal defects, and miscarriages. So sperm vacuoles has been this been studied till now. Okay, and how to select sperms? Sperms are graded into four grades. Okay, grade one, grade two, grade three, grade four, and grade one and grade two sperms can be injected. Okay, so if you see properly, spermatozoa, if there is normal forms and there is no vacuoles, that is grade one. If they're normal forms and the maximum there are only two small vacuoles, only 1% or something like two small vacuoles, that is grade 2. Okay, so these grade 1 and grade 2 sperms, they are best sperms. We can use those sperms. Coming to grade 3 and grade 4, that is class 3, class 4. In class 3, there are normal forms. By look, it is normal, but at least one largest vacuole and more than two small vacuoles. Small vacuoles are also there, and one big large vacuole also there. That is grade 3. Grade 4 is abnormal forms. They are properly abnormal and they are vacuoles also. Okay, that is grade 4. So grade 3 and grade 4 are not that useful. Okay, see, now I'm telling you now, in ICSI manipulator, if you see at 400 times magnification, it is looking that, so this is an ICSI people. Okay, to the visible eye, sperms are looking very good. But if you're doing on 8x for IMSI, 8000x times, if you see, there are nucleus vacuoles, abnormalities. Even in the separate sperms only, there are abnormalities and all. Okay, that is the difference. Coming to magnetic activated cell sorting. So, na, in adult testes, spermatogenesis creates so many millions of sperms. है ना? So, these sperms are largely controlled by apoptosis. Okay, that's why if you see sperms in semen sample, they're abnormal. Some sperms are abnormal forms, right? Right. It is because of apoptosis. But apoptosis is more than there will be more abnormal sperms, and these abnormal sperms should be killed by means of apoptosis, right? If the mechanism is defective, then abnormal sperms will be producing more. Mm, so, so multiple sperms show signs of apoptosis once ejaculated. Mm, so these sperms still have potential. Even though they have signs of apoptosis, they still have potential of fertilizing the oocyte, but outcome of embryo is questionable. Okay, that is why we go for MC. In this only we'll go for MC. This comes into picture. Okay, so MC, in MC there is a we can go for magnetic activating cell sorting device. This sorts the apoptotic sperms from non-apoptotic ones. It uses annexin 5, which has an affinity for phosphatidylserine. Okay, so see, good sperms means non-apoptotic ones have phosphatidylserine in them. Okay, now we'll keep annexin 5 in each and every chamber. If they're good sperms, then this annexin 5 go and bind to the phosphatidylserine. That means they're good sperms. Then we can select that. So in that way. So I am expecting this question. So they'll give this picture and they'll ask what is that, whether it is microfluidics or what is that. It's a MAX, that is magnetic activating cell sorting device. Okay. Application of max in IVF is it improves the fertilization potential. Obviously, then we are selecting a good sperms, right? And it improves the cleavage and pregnancy rates. It reduces the DNA fragmentation. Obviously, we are selecting the non-apoptotic ones. There will be no DNA fragmentation thing, and it also improves the live birth rate. Okay. Microfluidics. Next is microfluidics. It is also exciting technology where we can select good sperms. Okay. Mm, DNA, see, even the motile percentage has been increased. Okay. Um, so while doing ICSI, before ICSI, if you go for microfluidics, then we can easily separate the very good sperms. Okay, and this was the what you can say question in last INSET exam. They gave an image where they have like this rounds, and these rounds are being connected like this. Okay, and they gave options as max, microfluidics, and others, and that is answer is microfluidics. If you see microfluidics, you'll keep semen in this, and there are so many media, and motile sperms are going to here, and the outlet base sperms are going to the outlet. Okay, and see, these are dead sperms, and these are live and potential sperms. They're going into this chamber. Okay, that is microfluidics, and this helpful before going for ICSI. Okay, so conclusion of this chapter is Even though the technological advances are occurring at the rapid pace, the biggest challenge that still remains is justifying these technologies and proving their efficacy with long-term data to support. So there are different methods, but now even then that we are trying to prove that this is the best technique to select the sperm in that way. Okay. That's about this chapter. I'm expecting max image this time. Microfluidic image are already being given, and grade 1 and grade 2 sperms are very good sperms for fertilization. And yeah, okay. If you see this image, uh, you can find that sperm is bound to the hyaluronic acid. Okay, while doing PIXI, the sperms are bound to hyaluronic acid. Okay, that that's about how to select the sperm. Next chapter is now till now we learn about male thing. Now we learn about female. Okay, what are the different methods of ovulation stimulation? And after ovulation stimulation, you started ovulation stimulation to a person, and she's poor responders, and how to handle that kind of poor responder. That's it. Okay. Next we learn. So I'll tell in next video. Okay, about ovulation, female kind. Okay. Thank you.